Hi, I'm Kelly McCann. I'm a breast medical oncologist at University of California in Los Angeles, and I'd like to tell you about a clinical trial called Bria-ABC, which is a phase 3 vaccine trial for metastatic breast cancer patients. So first, what is the purpose of medicine clinical trials? The first purpose is to first demonstrate safety, do no harm, and the second is to demonstrate efficacy. The reason we do clinical trials is for consumer protection. The FDA was established in 1906 by Theodore Roosevelt to prohibit interstate transport of adulterated food and drugs and to prohibit false and fraudulent claims of curative or therapeutic effect through an amendment in 1912. At that time, there were things like The Jungle, if you've ever read that book, where food was being adulterated in a way that made it not safe for consumption, but it looked okay. And so this is why the Food and Drug Act was a very important consumer protection. In 1938, the Food, Drug, and Cosmetic Act was passed by Franklin Roosevelt. This included a pre-market safety review of new drugs and therapeutic devices, established regulatory standards, and gave the act a little bit more bite. It authorized factory inspections and enforcement power. There were a couple of drugs that I'll use as an example. One was sulfanilamide, which is basically antifreeze, and it was being used for people with colds, and patients were dying because they went into kidney failure. There was also another drug called Radiothor, which was radioactive water that patients would drink, and that was supposed to improve their vitality, but it ended up harming people. In 1951, the Durham-Humphrey Amendment was passed for prescription-only drugs, drugs that require the supervision of a medical professional, in addition to allowing for post-market recall of drugs. A couple of the drugs that have since been recalled, heroin, which was a Bayer product, and then this liquid bread that was marketed toward nursing mothers and their children, which was malt liquor. In the 1950s, the National Cancer Institute funded screening of synthetic compounds and plant extracts for anti-cancer activity. And this is how we came up with some of the chemotherapy drugs that we use today. In 1962 to 1964, the Pacific yew was found to have an extract that was cytotoxic. It killed cells in a petri dish. In 1966, the agent paclitaxel, which is also called Taxol, was isolated. Taxol then went through clinical trials, and in 1992, the FDA approved paclitaxel. So if you're a breast cancer patient, you probably have had Taxol if chemotherapy was recommended, or some version of Taxol, because this is one of the best drugs that we have for breast cancer. And if you've had that medication, you also know that it is very toxic. So how does the FDA decide whether or not they're going to approve a chemotherapy? It not only has to be safe in the hands of somebody who's trained to use it, but it also must be effective. When we're looking for new medications, first we start in the laboratory. Preclinical research goals are to find and make new therapies. A drug may be determined to be safe to give in animal models, but ultimately we need to test it in human beings. In preclinical research, we might be looking at how is this medication working? We need to come up with an explanation for how that treatment works before we're going to use it in human beings. If promising, a sponsor, which might be a drug company or a lab or a government institution, will file an investigational new drug application with the FDA to ask permission to study the drug in humans. Phase 1 clinical trials are meant to determine, is this drug safe to give humans? The number of patients is usually about 50 or so, and they can be patients who have progressed through all standard therapies for whom no effective therapy exists. So we don't put patients on phase 1 clinical trials, which are safety trials, until we've exhausted a lot of other options. The clinical trial can go on for months to years, and the main goals are to find out how to give this drug in a safe way. How should the treatment be given? Should it be IV or oral, etc.? We determine a safe dose by dose escalation. We start very, very, very low, give that to 3 people usually. If they tolerate it well, then we go up on the dose and up until we hit some kind of maximum tolerated dose, which is usually that a patient developed a toxicity that we don't want our patients in the future to ever have again. There are also pharmacokinetic studies. When we give a medication, we want to know, how is that drug absorbed? How's it metabolized? How does it spread through the body? But most importantly, we want to know, when we give that drug to a person, what kind of adverse effects are they going to get? And so all of those effects are logged very, very carefully. Even though efficacy is not the goal of a phase 1, we're excited if a drug works, and we want to know why. When we're logging adverse events in any of our clinical trials, this is basically a review of systems. An adverse event is any unfavorable or unintended sign, symptom, lab, or disease that's associated with a medical treatment or procedure that may or may not be considered related to the medical treatment or procedure. So if you, for example, are on a clinical trial and you developed a cold, we're going to record that as an adverse event, even though that adverse event wasn't secondary to the medication that you were receiving. The National Cancer Institute specifies how we grade side effects in the common terminology criteria for adverse events. If you look at this chart, you'll see a grade of 0, 1, 2, 3, 4, and 5. If a patient is having no side effects whatsoever, then any possible symptom is going to have a grade 0. That's the patient's baseline. If someone is having mild side effects, that's a grade 1. Up to 2 is moderate, 3 is interfering with daily life, 4 is life-threatening or disabling, and 5 is death secondary to a side effect. So if you're reading a clinical trial paper, there will always be a chart in that paper that will tell you how many of those patients got those side effects, and then will grade those side effects as well. If a patient is experiencing a grade 3 or 4, or God forbid, a 5 side effect, that's going to be called out in the paper so that you know about that. And in the future, if that drug does pass FDA, we will know as oncologists to monitor for those effects very carefully. An example of having a graded side effect is a side effect like diarrhea. So a grade 0 diarrhea is same number of stools as baseline. A grade 3 diarrhea is 7 or more stools a day over baseline. Maybe IV fluids were needed for more than 24 hours. And 4 would be disabling or life-threatening diarrhea, in which a patient would end up in the hospital and need support. Prescription pamphlets list all of the adverse events that occur in a clinical trial, but not necessarily with the percentage of patients who experience that event. And so you must interpret patient pamphlets with some caution. Phase 2 clinical trials are for, does this treatment have a therapeutic benefit? It can be a few hundred patients, usually a very specific tumor type like breast cancer. The length of time might be months to years. All patients in the clinical trial typically receive the new therapy. And the goals are to monitor for adverse events, further define the toxicity, determine if the treatment affects cancer, which is based on symptoms, frequent imaging, and may or may not include tumor markers. And then to be studied further, we want the response rate of the new treatment to be promising. How do we determine response to treatment? This is usually done by RECIST criteria. We look at the same lesions in a patient, lesion being a spot of cancer, and we measure them on repeat imaging. So for example, on the CT scan on the left, you can see with the yellow arrow, the white arrow, and the red arrow that there are several big white splotches. Those are cancer metastases. And then over time with serial imaging on the right, you can see that those same little nodules have shrunk significantly. So when we're looking at the serial imaging, we want to see how much did those target lesions shrink. If the target lesions all disappear, that's called a complete response. If there's a 30% decrease in the sum of those target lesions, that's a partial response. And if there's an increase by 20%, we call that progressive disease, and the patient is on to the next therapy because it's no longer working for their cancer. It's also important that stable disease, which are small changes that don't meet criteria for complete response, partial response, or progressive disease, are also very important, especially in slow-growing cancers where you don't see shrinkage or growth very quickly. Phase 3 trials are meant to determine, are these results that we see with this new medication better than our standard of care at that time? And this can be a big trial with hundreds of thousands of patients. patient population is usually a specific tumor type, but usually we want a very diverse patient population. If we're going to roll a medication out across the US or across the world, then we want to see a lot of different types of patients, in both in terms of ethnicity, age, and sometimes gender. These trials can go on for months to years. The design is usually randomized, meaning that a patient might have a flip of the coin get randomized to either the standard of care at that time or the new treatment. They can be double blind as well, meaning the patients and the physicians don't know which arm they're on. There's never a time in an oncology clinical trial where the patient gets absolutely nothing, because that would be unethical. A more likely scenario might be that a patient is on a standard drug that we know treats cancer, and maybe a second drug is added on to see, does that enhance the therapy that we are looking at. The goals of the phase 3 are to compare the new treatment with the standard of care in terms of efficacy and side effects. Surrogate endpoints for overall survival can include the time it takes for the cancer to grow again, as well as if you're in the curative setting, whether or not cancer recurs. And of course, as with all clinical trials, we're always monitoring for adverse effects and recording those. Our outcome measures can be overall response rate, which is adding up the complete remissions and the partial remissions, a clinical benefit rate, which is defined by complete remissions plus the partial remissions plus stable disease, progression-free survival, which is the length of time until the cancer grows again, disease-free survival, which is the length of time until disease recurs, and then overall survival, which is time until death. If a new therapy doesn't increase overall survival, it may not be FDA approved, or FDA approval may be withdrawn. And now to look at the mechanism of action for the Bria-IMT. This is a cell line of hormone receptor negative, HER2 positive breast cells that have been modified to secrete GM-CSF, which is a protein that stimulates the immune system. It also has been modified to be able to present cancer proteins to your immune system, and finally to stimulate cancer-killing immune cells. Then these cells are irradiated to kill those cells to create a vaccine. And this kind of strategy has been used for a lot of our vaccines, where you take something that was lethal and then you irradiate it so that it is killed, but it still has all those proteins associated with it, so that your immune system can recognize those cells and then go on to kill any live cells that might be in your body. In the phase 1/2 Bria-IMT clinical trial, which included 54 patients, the average age of patients, or the median age, was 61. The patients were, in general, very healthy, capable of still working, and they had been heavily pretreated with different types of therapies. For example, antibody drug conjugates, immune checkpoint inhibitors, CDK4/6 inhibitors. This clinical trial included patients from all types of breast cancer, and so the types of therapies that they had received beforehand really varied quite a bit. This is the most important thing to come out of a phase 1 trial, which is our table of adverse events. So what happened to those patients? In the bold, under a related column, these are the adverse events that the people running the trial thought were due to the medication itself, to the vaccine. 22% of patients, for example, had fatigue. About a third of them had an injection site reaction, which is very common at the site of a vaccine. Some patients had some nausea, diarrhea, or constipation. Some patients got a rash. There were also patients who had fevers, which sometimes happens with a vaccine clinical trial. And then these are some of the results. If you look at the blue bar, that's the percentage of patients who had an overall response, who either had complete disappearance of all of their metastasis sites, or they had a partial remission, which means that those sites shrunk down. About 10% of patients overall in this trial had an overall response rate. And in the best clinical response, clinical benefit, about 55% of patients had some level of benefit from the therapy, including stable disease, which for slow-growing cancers might be a win as well. There were also interesting results that showed that maybe in looking at this imaging on the left going to the right, you can see that the small little yellow dots, that's cancer. And then after giving this vaccine therapy, some of those yellow spread, that's inflammation. So we think that maybe the Bria-IMT combination therapy can induce the immune system to come into metastatic breast cancer and fight off those cancer cells. So the way that the phase 3 Bria-IMT regimen is given is in 3 big phases. 1, enhance the immune system. 2, give the vaccination. And then 3, stimulate the immune cells at the vaccine site. So how is this done? Well, to enhance the immune system, researchers have found that low-dose cyclophosphamide can enhance an anti-tumor response by suppressing normal cells that protect cancer cells from destruction. There's also an immunotherapy called retifanlimab, which enhances the immune response by unmasking cancer cells to the immune system. Those 2 medications are given before the vaccination. After the vaccination, which is performed on the thighs and the upper back, so that there are places that a investigator can measure, the patient is also given a little bonus inoculation with interferon, which is a signaling molecule that can activate immune cells at the site of infections and tumors. The clinical trial is designed to randomize patients to either the vaccine with the immune checkpoint inhibitor, or to randomize them to the standard of care, which is physician's choice of chemotherapy. And there are a lot of different types of choices, among them carboplatin, taxane, capecitabine, gemcitabine, numerous choices. In this clinical trial, in the blue box on the left, you can see that some of the inclusion criteria include metastatic breast cancer. The patients, even though are all types of breast cancer eligible, are stratified by hormone receptor positive and HER2 positive. Stable brain metastases patients are allowed. Stable endocrinopathies, like having diabetes, for example, are allowed in the clinical trial. But patients with autoimmune diseases that require immunosuppressants are excluded. And that's because when we're giving a vaccine, if you're already on something that is immunosuppressant, then we don't think that the vaccine has much of a chance to work. The primary endpoints of this clinical trial are to look to see overall survival, but secondary endpoints are to see, does the cancer grow again? How many patients responded? How well did they respond? Safety, as always, quality of life. In the inclusion criteria for any clinical trial, it's important to note just how the patient population is defined. So in this clinical trial, all breast cancer patients are allowed, brain metastases are allowed, and measurable disease, meaning something that you can look at on an image and measure in terms of centimeters, is not required. So that's very important, because a lot of times patients with brain metastases are excluded from clinical trials. A lot of times bone-only metastatic disease is also excluded. In any clinical trial, there are requirements for prior treatments, or there may be a requirement that the patient had had no prior treatment. In this one, depending on the type of breast cancer, those patients had to have had appropriate targeted therapy before they're eligible for the clinical trial. For example, if a patient has a hormone receptor positive tumor, they must be refractory to hormonal therapy, as demonstrated by progression on at least 2 hormonal agents. For the HER2 positive patients, they must have already had 2 prior HER2 targeted regimens. And for triple negative breast cancer patients, they must have had progression on at least 2 prior chemotherapy regimens. One of the important parts of determining whether a patient is eligible for a clinical trial is also to make sure that the patients who are enrolled are healthy. We don't want to harm anyone by putting someone who is unhealthy or has some kind of organ dysfunction onto a clinical trial, because we could cause harm. So patients must have a performance status of at least being able to get up, come into the clinic, walk around, do light housework. Finally, there are always criteria to protect patients who may become pregnant. And so women and men of childbearing potential must agree to take precautions to avoid pregnancy. In all clinical trials, there is a patient bill of rights. You have rights in any clinical trials. Cancer clinical trials never include a placebo alone option in the metastatic setting because that would be unethical. We don't put patients on clinical trials and give them nothing and just watch their cancer progress. We only put patients on trials where we are giving you something that we are hopeful and have evidence for will help treat the cancer. Prior to the initiation of a clinical trial at an institution, there are multiple safeguards in place, including an institutional review board to ensure ethical conduct and participant safety, an internal scientific review committee, and disease-specific researchers who try to make sure that that trial is worthy of study before it gets started. We have limited resources and we want to study things that are most likely to work. Other departments like radiology, pathology, and others also help to minimize risk by doing their own check on the clinical trial. In addition, the budgeting team negotiates to ensure patient costs are covered, which includes safety measures and making sure that if something happens, that the clinical trial or insurance is going to cover those costs to keep that patient safe. Patients have a right to informed consent about the trial, which includes purpose, procedures, potential risks, and benefits. Informed consent has to be signed before any screening procedures must take place. Pre-screening means blood tests, imaging, cardiac evaluations, and exams that are before the trial so that we can evaluate that patient to make sure that they're safe to go on a clinical trial. This pre-screening might take up to 28 days from the time of signing informed consent until the first dose of medication. So it's important to know that before you sign consent that there's going to be a little waiting period before you can get started while we assess for safety. Patients have a right to withdraw from a trial at any point and for any reason without consequences, judgment, or affecting their health care. Patients have a right to information about study procedures and adverse reactions and a right to their own data. Adverse reactions might become known during the trial, in which case a revised informed consent is required to allow a patient to determine whether or not they want to continue on the trial. In a double-blinded clinical trial, the one piece of information that patient and physician agree to not have is which treatment the patient is receiving, and that would be more typical of a phase 3 trial. And then finally, patients have a right to confidentiality and privacy. All data that's transmitted to the sponsor of a clinical trial is de-identified. So the person is given a number usually so that nobody outside of your doctors at the institution for the clinical trial know who you are. Does insurance pay for clinical trials? If you are part of a clinical trial, your own insurance coverage funds a lot of the standard of care procedures, such as routine imaging, blood tests, doctor visits, and medications. But the sponsor of the clinical trial, which might be a pharmaceutical company, academic center, NIH, or cooperative group, pays for the parts of the trial that are not standard of care, which might need the medication under investigation, medications needed to prevent side effects of that medication, extra blood tests, extra imaging, and extra doctor visits conducted for your safety and to help us understand the medication under investigation, like how it's metabolized by your body. How do you find a clinical trial? The first step is to ask your oncologist. Cancer care is collaborative. If a clinical trial is an option, your oncologist can refer you to a center if they don't have a clinical trial for you themselves. Utilize cancer advocacy groups like American Cancer Society, Susan G. Komen, and the Breast Cancer Research Foundation for information and support. And finally, do an online search through clinicaltrials.gov registry for clinical trials in your area or places to which you're willing to travel for a trial. One important caveat for clinical trials is that we set them up at a particular institution. My clinical trials are at UCLA. I trust my team. I'm responsible for the patients. That clinical trial might not be available at a more convenient location for you, and you might have to travel. So keep that in mind when you're looking at clinical trials and look at the schedule very carefully and try to find out if that schedule is going to fit into your life. And finally, if there are any questions, I work in UCLA breast cancer research, and my email is as follows. Thank you very much.